Hello everyone, welcome to Visionary First Step YouTube channel. Today I am going to explain you the topic sexual reproduction in flowering plants. See, whenever the term sexual reproduction comes, we will try to understand the basic mode of reproduction which is present in the organisms. There are mainly two types of reproduction. One is asexual mode of reproduction, another one is sexual mode of reproduction. Now, we have studied that in our earlier classes, asexual mode of reproduction where vegetative part of the organism will get separated simply by fragmentation, and that fragment will grow into a new organism. But in higher plants, we observe specialized structures which help in sexual mode of reproduction, where we observe interaction of male and female gametes, and fusion of these gametes give rise to formation of zygote, and zygote develop into embryo, embryo develop into young ones. So this is the sequence how the sexually reproducing organisms are produced. Now, when you look at this slide, what do you feel actually? So this is one beautiful hibiscus flower. Now, why these flowers are produced in flowering plants? These flowers are the primary structures which help in sexual mode of reproduction in angiosperms. So what and all the whorls are present in a flower which helps in sexual mode of reproduction? Outermost whorl we call it as sepal, and inner to that petal is present. See, these two are actually accessory whorls. Now, why they are present actually? Sepals which help the flower in bud condition, so that the floral bud will not undergo any type of damage. So the primary function of the sepal is to give the protection. Petals, as we all know, here you can see such beautiful petals are present, but why they are present? You know, it's a type of attracting the insects, or you can say pollinators. Angiosperm, they mainly require pollinators for the pollination, which will finally lead to the sexual mode of reproduction. So to attract, they have a very beautiful petals. In other words, it is also called corolla. Okay, corolla is for petals, calyx is for sepals. So these two structures in the floral whorl, they indirectly help in sexual reproduction, but directly androecium and gynoecium involve in sexual reproduction. The common name is stamen, which is the male reproductive structure. In taxonomy, we use the term androecium. Similarly, for female reproductive structure, we call it as pistil, and in taxonomy, these structures are called gynoecium. Okay, so if we observe, there are four floral whorls. In that, two are accessory whorls and two are essential whorls. Accessory whorls are sepal, and in taxonomy we use the term calyx, which is the outermost, green in color, and inner to that petal is present, which is the common word we use to represent the colorful part of the flower, and corolla is the term which commonly used in taxonomy. Now, essential whorls which mainly involve in sexual reproduction, that is stamen is the common name. Male part is also known as androecium in taxonomy. Female reproductive part is pistil. Sometimes we also use the term carpel. See, pistil is only representing female reproductive part, that is stigma, style and ovary. Later we will study in detail. Sometimes we use the term carpel in taxonomy. The only reason is flower may have more than one female reproductive structure. At that time, we cannot use the term pistil, so we use the term carpel. Now, gynoecium is the term used in taxonomy. Okay, again we'll just look at this flower. Just observe, this is the stalk of the flower. What we can call this stalk of the flower? In first year we studied this. This is called pedicel. Okay, so this part is pedicel. Above there is a thalamus. Here you can see this is the thalamus part, receptacle part from which floral whorls will originate. Outermost here you can see this is not calyx, okay, it is epicalyx, observed in hibiscus. Okay, next comes calyx, common name is sepal, and then corolla. Here you can see the corolla, they are all fused actually, gamopetalous condition, and here you can see the gynoecium part. This is the gynoecium part, female reproductive structure, which passes through the tubular structure, in this case of hibiscus. Here you can see actually there are 5 gynoecium, they are all fused and passing through the funnel like structure that is formed by the fusion of all the androecium. That condition we call it as monoadelphous condition. Okay, so this is how we'll study a flower. Now tell me, who is not interested in flower? Everybody likes the flower. Flower is a fascinating organ. Why? Because for any aesthetic occasions we use the flower. To congratulate someone we use the flower. To celebrate any festivals we use the flower. For daily puja we use the flower. Women in India, they used to wear the flower daily because they have a beautiful fragrant. So flower is a fascinating organ for all the organisms, for even birds, animals, insects. They all like flower because plants, they provide certain kind of rewards when someone visits the flower, either in the form of fragrance, or in the form of nectars, or in the form of beautiful colored flowers. So this is how flowers, they are the fascinating organ. Now this is again a typical flower showing different parts. Here you can see the thalamus. Above that floral whorls are present. Outermost is the sepal, and the large colored showy part is the petal, and male reproductive structure is androecium and female reproductive structure is gynoecium. Now coming to the main part of the topic. Here we segment this topic into 3 main parts. One is pre-fertilization event, fertilization process, and post-fertilization events. Now tell me, what is fertilization actually? Fertilization is nothing but the fusion of male and female gametes. Now the process of formation of male and female gamete, we call it as pre-fertilization structures. See, for the formation of male gamete and female gamete, there should be a specific structure in the plant which help to bear the male gamete and female gamete. So initially we study the structure which helps in male gamete, nothing but the androecium, and a structure which helps in production of the egg, nothing but the gynoecium. Okay, that we will study in detail in the pre-fertilization structure and event. Event in the sense how these structures, androecium and gynoecium, they produce pollen grain and the egg. That we will study in detail. Fertilization in the sense, now once the pollen grain are produced, either by the wind current or by the insects or by the different mode of transport, how they reach the stigmatic surface and what happens on the stigmatic surface, the pollen grain, they germinate and they reaches the ovule where it fertilizes with the egg. Okay, this process we call it as double fertilization, that we will study in detail. It's a very interesting topic. That is about the fertilization. Post-fertilization in the sense, what happens after fertilization? Zygote is formed. What happens to zygote? Zygote develops into embryo. What happens to embryo? Embryo is present in the seeds. That means seed is nothing but the what? It is the fertilized ovule. Where these seeds are present? Inside the fruit. Then what is fruit? Fruit is nothing but the fertilized ovary. So this is how we study the structure of fruit, structure of seed, structure of embryo, all those things in the post-fertilization events, after fertilization. So this is the main segments of sexual reproduction of flowering plant topic. So let us start with the pre-fertilization structure and events. Hope you have a little idea about the floral primordium. You know what is leaf primordium? Part of the shoot which has the destiny to become leaf, they are called leaf primordium. Similarly, once the plant attains certain maturity, the part of the plant will decide this part will become a flower. So there are so many hormonal changes takes place in the plant body, like auxins are produced, cytokinins are produced, then some of the hormones specifically for flower production are produced. These hormones make certain changes in the plants, and part of the plant will destined to become a flower, and that part we call it as floral primordium. This is very important term. This floral primordium later develop into a inflorescence, and this inflorescence later produces the flowers. Okay, now what and all the hormones are present that already we studied in first PUC, plant growth and development. Mainly the auxin, cytokinin, especially cytokinin and ethylene, they play a very important role in production of flowers. Now inflorescences are formed as I told, and then they bear the flower. In flower we observe male reproductive part, that is androecium, and female reproductive part, that is gynoecium. Okay, now let us study in detail about the male reproductive structure. The common name is stamen, and inside the stamen what is present? Microsporangium. This microsporangium contains pollen grains. So this all together nothing but the male reproductive structure. Now how many of you have observed the flowers? Everybody, right? So we just observe the flower, but observe in detail how these whorls are arranged. Just observe the stamen, from where it is originating, whether it is originating from the thalamus, whether it is originating from the petal, or which type of stamens are present. They are long or short, or the lobe of the stamen is kidney shaped, or is it is elongated. So there are so many types of stamens are present. It is not same in all the flowers, and it varies from flower to flower. Even the number of stamens varies, length of the stamen varies, shape of the stamens varies, number of pollen grains varies. So it is not same in all the flowers. Now can you tell me why it varies? It mainly depends on type of pollination, because let us consider a type of flower which is very small, inconspicuous, and it is not attracting any type of insects or pollinators for the pollination. How will it reaches its male gamete to the site of female gamete, right? So at that time it produces numerous pollen grains, and then pollen grains through the wind or water reaches the stigmatic surface, pollination takes place, right? Now how these stamens are attached to flower? They have a long and slender filament which originate either from the thalamus or from the petal. If it is originating from the petal, then the condition is called epipetalous condition. Okay, so this long slender stalk called filament and a terminal generally bilobed structure is called anther. In the next slide I will show you the picture. Here like this slender filament is present, here lobed anther is present, and filament and the anther are connected by the connective. Okay, this is the anther. This is filament. Now, here there is one very important statement which usually asked in competitive exams. What is that actually? The proximal end of the filament is attached to the thalamus or the petal of the flower. Why it is given proximal? Right? So it is referring to the thalamus. Okay, that is why they have given proximal end. Sometimes in competitive exam, what they will do, instead of giving proximal end, they just change one word and make it distal end. So be careful while reading this statement. Proximal end of the filament attaches to the thalamus. That means in the filament, this is proximal and this is distal. Suppose somebody frame the sentence like this: the filament of the anther in the distal and anther is present. Whether this statement is correct or wrong? Absolutely right, because they have already mentioned that proximal end is attached to thalamus, then anther should be present on the distal end. So be careful about this particular statement, because in one of the NEET exam they asked this. Okay, very important. The number and the length of the stamens are variable from species to species. Take different flowers, take hibiscus flower, take jasmine flower, or take any of the flower of your garden. Just observe the stamens, they are not same, they are different. Now, a typical angiosperm anther is bilobed. This is very important. And they have mentioned that typical angiosperm anther lobe. That means there are chances this lobe is single lobed. There are chances it may have two or more lobes. But in common, in most of the plants, we observed bilobed anther. Bilobed in the sense what? Now, if I say, see, this is one anther, the lower part is the filament, upper part is the anther. If you just observe that, it is like this with the two lobes and they are fused. Okay, so that is called bilobed anther. What is there inside the bilobed anther? That we'll see. Okay, often longitudinal grooves run lengthwise and separating the theca. We'll see what is theca in this slide. Just observe here. See, this is one anther lobe. It's clearly observed that this is one lobe, this is another lobe. Bilobed condition, or you can say dithecous condition. Here in the transverse section, we can clearly observe that these are the two lobes. Now each lobe is having two chambers like this, right? So bilobed dithecous condition, each lobe is having two chambers. That is why they are called dithecous condition. Suppose in competitive exam, if they ask how many microsporangium is present, what you will say? How many are there in one anther? In one anther it is 4 microsporangium. Nothing but how many pollen sacs are present? 4 pollen sacs are present, because microsporangium develop into pollen sacs. We'll try to understand words one by one. The bilobed nature of the anther is very distinct in the transverse section of the anther. So only when we take the transverse section and observe it under the dissection microscope, we'll get to know the bilobed condition. Okay, clearly. Now, the anther is a 4-sided structure consisting of 4 microsporangia located at the corners, 2 in the each lobe. Here we can clearly observe the microsporangia. See, try to understand by the term microsporangia. Micro is something representing the gamete which is smaller. That is why the term microsporangia. Microsporangia is the structure inside the microspores are present. That means it is something related to male reproductive part of the flower. Now, in this anther, The microsporangium is very clear only after taking the section. Now, where they are present at the corners. If you observe the bilobed anther and take a transverse section, in the corners you observe the chambers, and inside the chamber what is present? Microsporangium is present. How many present? Each lobe consisting of 2 microsporangium. Now this microsporangium matures and develop into pollen sacs. Now why the term pollen sac? Because inside that pollen grains are produced. How they are produced, that we will study in detail in microsporogenesis. Genesis something to related to the formation. Clear? Now the microsporangia develops further and becomes the pollen sac. There is a reason to give this statement in a different color. This question is asked once in examination. Which part will directly develop into a pollen sac actually in the male reproductive structure? That is microsporangia. Okay, they extend longitudinally all through the length of an anther and are packed with the pollen grains. So if you observe here, they are present all along. It is not confined to a one place of the anther. You cannot say it is just present at the base of the anther, or it is located at the tip of the anther, it is located only in the middle of the anther. Not like that. This microsporangium run all along the anther. Is it clear? It is present fully inside. And inside that there are specialized cells. They are called sporogenous tissue. And they develop into microspore mother cells. And these microspore mother cells are also known as pollen mother cells. And these pollen mother cells undergo meiosis, very important. Meiotically, they produce the pollen tetrads, and pollen tetrad further develop into pollen grains. So I will explain that in detail. Hope this is clear. These microsporangium, they bear the pollen grains. Now just look at this diagram. This is very, very important part. What is there inside the male reproductive structure? That means if you take the transverse section of anther, what and all layers are present, and the innermost layer, what is there, and what is the function of all these tissues, that we will study in detail. Structure of the microsporangium. Just observe here, this is the generalized outline of the anther, and this is one part enlarged. Here you can see the same structure, but it is in the dehisced condition. That means after maturation, wall of the anther will dehisce and help in release of the pollen grains to the outside environment. Now, so this is the young anther. Just observe, this is the young anther. When you observe this enlarged part, the outermost layer, here you can see the outermost layer. This is the outermost layer, and we call it as epidermis. Epi means outermost, dermis means layer. Next to the epidermis is the endothecium. Again, one more layer is present, a layer of cells. When you look at all these cells, they are simply a parenchymatous cells. They are compactly arranged, not tightly arranged. There is an intercellular space. Okay. So endothecium is present just below the epidermis. Please remember the sequence of arrangement of these cells, because sometimes a sequence based question is asked on this topic. The outermost is epidermis. Next to epidermis is the endothecium. Next to endothecium is the middle layer. Here there is one important aspect. Epidermis is single layered. Endothecium is again a single layered, but middle layer is multi-layered. Okay, so here you can see the middle layer with the many layer of cells, and the innermost, here you can see, so these are the tapetal cells. Tapetum, actually in Latin tapetum means just a cover. Okay, so why here tapetum term is used? Because it is covering the inner pollen sac, so the term tapetum. There is one interesting aspect about this tapetum. All the cells, epidermis, endothecium, middle layer, they are all the type of cell where only one nucleus is present. But tapetum is a specialized type of cell. When the time they are formed, they have two or more nucleus. Now question comes, why they have two or more nucleus and what is their role? Epidermis, endothelium and middle layer, they mainly involve in dehiscence. Once the anther is completely dehydrated, pollen grains are fully formed and they are mature. These three layers, they absorb water from the atmosphere and they rupture and pollen grains are released. So that is the function of three layers, epidermis, endothelium and middle layer. Then what is the role of this tapetum actually? I just said tapetum in the sense in Latin word, Latin it is meaning is just a cover. So this covering cells, tapetal cells, they act as a nursing cells or it nourishes the developing pollen grains. So it has to do multiple activities. So they have two or more nucleus which involve in helping those activities. Okay, and they have a dense cytoplasm compared to other cells. Now, inner to this is the sporogenous tissue. Here you can see this part actually consisting of sporogenous tissue. What is the role of this sporogenous tissue actually? Very interesting part. Sporogenous tissue are actually dividing cells, just like a meristematic cells. These sporogenous tissue at one particular time, they divide mitotically and increase their number and they become pollen mother cell, or you can say microspore mother cells. These microspore mother cell later they undergo meiosis and produces the pollen grains. Now the same thing is explained here. The role of epidermis, endothelium, middle layer and tapetum. Again, remember the sequence properly because sequence based questions can be expected from this topic in competitive examination. Okay. Here I just included a small explanation related to this. It is not there in NCERT, but you should know what it is. Tapetum possess dense cytoplasm and generally have more than one nucleus. To support their vital role in nourishing developing pollen by boosting the metabolic activity, increasing gene expression and facilitating rapid synthesis and transport of nutrients like sugars and proteins essential for pollen wall formation and viability. So these are the different functions done by tapetal cells. Okay, so it is just a addition of information to tapetum because you need to remember this tapetum, a most important aspect in studying the transverse section of anther. Okay, so can you think of how tapetal cell could become binucleate? Sometimes they will ask this question. So this is mainly because of endomitosis. Very important. Here mitosis takes place, nucleus undergo duplication, that means karyokinesis takes place, but which is not followed by the cytokinesis. If cytokinesis takes place, this will become a two cell, but it will remain as a single cell but has two or more nucleus. That is possible mainly because of endomitosis. Okay, when the anther is young, the group of compactly arranged homogeneous cells called sporogenous tissue occupies the center of each microsporangium. As we studied, in an anther, two lobes are present, corner of the lobes inside consisting of 4 chambers, and that is called microsporangium. And in the core of the microsporangium, we observe a special type of cells and they are called sporogenous tissue. Why they are specialized cells? Because these sporogenous tissues, during the time of microsporogenesis, they become microspore mother cell or pollen mother cell and helps in the formation of pollen grains. Is it clear? Okay, let us move on to microsporogenesis. Microsporogenesis is very interesting process. So what happens here? This all takes place inside the anther. Okay, we studied epidermis, endothelium, middle layer, tapetum, and after reaching the center of this microsporangium, there is a group of tissue and they are called sporogenous tissue. What is the main role of this sporogenous tissue? They undergo mitosis and increases their number and become microspore mother cell, or in other words we can also say pollen mother cells, and they undergo meiosis and produces the pollen grains. Okay, so in other words you can simply say sporogenous tissue, they undergo meiosis and produces the pollen grains, in order to just have a sequence. No need to explain that sporogenous tissue undergo mitosis and increases their number and becomes microspore mother cell. You just can say that sporogenous tissue undergo meiosis and produces the microspore tetrad that separates and later become the pollen grains. Now, what would be the ploidy of the cell of the tetrad? Do you know what is ploidy? Ploidy is nothing but the number of chromosomes in a cell. Now, when the cell which is diploid undergo meiosis, how many number of chromosomes will be retained in the daughter cells? That is haploid. That means half of the number of chromosomes are retained in the microspore tetrad. So the ploidy of the microspore tetrad is haploid. As each cell of the sporogenous tissue is capable of giving rise to a microspore tetrad, each one is a potential pollen or microspore mother cell. That means don't get confused with this sentence. How this sporogenous tissue is capable of giving rise to a pollen grain and microspore mother cell. Not like that. The meaning of the sentence is something like this: sporogenous tissue is capable of giving rise to a pollen mother cell. Each one is capable of giving rise to a pollen mother cell. Similarly, each pollen mother cell is capable of giving rise to a pollen grain. That means what? Each sporogenous tissue is capable of giving rise to a pollen grain as well. Okay, that is the meaning of this statement. The process of formation of microspores from the pollen mother cell through the meiosis is called microsporogenesis. That is the definition. What is the definition? The process of formation of pollen grain from the microspore mother cell is called microsporogenesis. The microspores, as they are formed, are arranged in the cluster of 4 cells and they are called the microspore tetrads. Now imagine, inside the microsporangium, now sporogenous tissues are present. These sporogenous tissues later become a microspore mother cells. Now microspore mother cells undergone meiosis. In the first meiotic division, they formed dyad. In the second meiotic division, they formed the tetrad. Now in these tetrads, they immediately will not undergo separation. In the first phase, they are together and forming a tetrad, and that we call it as microspore tetrad. As the anther matures and dehydrate, the microspores dissociate from each other and develop into pollen grains. So the pollen grains are observed only after anthers are fully matured and when it is ready to dehyse and release the pollen grains. Okay, so inside each microsporangium, several thousands of microspores or pollen grains are formed and that are released with the dehiscence of anther. Now tell me why there is a need of production of several thousands of microspores or pollen grains. Because it is a chance factor. Nobody can assure that produced every pollen grain lands on the stigmatic surface and undertake the pollination. Some of them may be lost, some of them may be carried by the insects as a source of food, or some of them will fall off. So certain will reach and produces forms the function of fertilization. Similarly, in the case of human beings also, we all know that we produce millions of sperms to reach one egg. What is the reason? Because in that millions of sperms, most of will die in the movement or in the travel from the female reproductive tract to the egg. Similar things happens in plants also. So the number of pollen grains or the shape of the pollen grain, weight of the pollen grain and sticky nature of the pollen grain all will vary from species to species. Why? Because each and every plants are unique in their mode of pollination or fertilization process. Okay. Now let us go in deep and depth regarding the pollen grains. Here I included one picture. It is not there in your NCERT, but it is the photograph of the pollen tetrad. Just observe. It is just formed pollen tetrad. All the 4 pollen grains are together. All the 4 pollen grains are together in the tetrad form. So this is from the different species. These pollen tetrads, once they get separate, they become pollen grains. So here the pollen grain shown is not from one species, it is from the different species of plants. Now what happens to this pollen grain? Initially it is single, it has a single nucleus, that is haploid nucleus. Now this haploid nucleus, pollen grain, starts to develop a vacuole, and this nucleus undergo mitotic division. Very important. Haploid cell again undergo mitosis. During that time, it forms the asymmetric spindle. Very, very important character here. Usually cell division, at the time of cell division, what type of spindles are formed? Symmetric spindles are formed. But here, surprisingly, it forms the asymmetric spindle. The reason is the next forming cells are of not same sized. They are of not same sized, because one will become a generative cell, another one will become a vegetative cell. They both have nucleus, and one is small, another one is large. In other words, there is one beautiful sentence given in your NCERT textbook. What they say? Generative cell floats in the cytoplasm of the vegetative cell. See how beautifully they have given. The reason is, these vegetative cells, the nucleus which is help in formation of pollen tube. Okay, in other words, you can also say pollen tube nucleus. So which guides the pollen tube, and generative cell, it floats in the cytoplasm of the vegetative cell and reaches the female reproductive part. Okay, so that is why sentence is used. Here this picture is given in NCERT. This is the picture given in NCERT, and these 3 diagrams I have taken it from the different source. Hope this diagram is very clear now. Now tell me, how many cells are present in the matured pollen grain? 2 cells, one generative cell, one vegetative cell. Is it same in the case of all the plants? No. 60% of the plants, they release their pollen grains in the 2-celled stage. Remaining 40% of the plants, they release their pollen grain in the 3-celled stage. Now again a question, what is 3-celled stage? Very important. This generative cell further undergo mitotic division and form a 2 generative cell. These 2 generative cell has 2 male gametes, and which are responsible for double fertilization. One of the male gamete go and binds with the egg, and fertilization takes place. Another male gamete go and bind with the polar nuclei, and fertilization takes place. These 2 takes place together. That is why we use the term double fertilization. Now all the details regarding to pollen grain I explained. Once again they have given here. Pollen grains represent the male gametophyte. Please do remember, because there are so many terms here. Male gamete, male gametophyte. If pollen grain is the male gametophyte, then what is male gamete? Generative cell is the male gamete. Please remember, most of the time in competitive exam, they will confuse you with the microsporangium, microspore mother cell, then pollen mother cell. Similarly, male gametophyte, male gamete. So these are the some of the terms which creates lot of ambiguity or confusions in students. Make sure that pollen grain, remember, pollen grain contains male gamete. So pollen grain will be male gametophyte. The structure which bears the gamete are called gametophytes. So the pollen grain is gametophyte. Pollen grains are generally spherical, measuring about 25 to 50 micrometers in diameter. So sometimes they will ask the size of the pollen grain also. They have given ranges here, 25 to 50 micrometers. It is in micrometers. It has a prominent two-layered walls. Now we will move on to the detailed structure of the pollen grain. So far we just studied what happens to pollen grain after tetrad is formed. Now, what and all the changes takes place in the pollen grain before it will become mature, that we should observe. In the last topic, while explaining the tapetum, I explained that this tapetum helps in formation of different layers of the pollen grain in the later stage. Just observe this pollen grain. This is the inner part where cytoplasm is present. There are two nucleus. One is tube nucleus, nothing but the nucleus of vegetative cell. Another one is the nucleus of generative cell. Now this generative cell floats in the cytoplasm of the vegetative cell. To this particular pollen grain, internally it has intine, externally it has exine. Intine is a continuous wall layer, exine is not continuous, it is interrupted. Interrupted with what? Germ pores. Why these germ pores are important? Through which pollen tubes are formed. Imagine now pollen grain in the wind reaches the stigmatic surface. Now it has to germinate the tube, then only it can reach its male gamete to the female reproductive structure. That's why germ pore is very important structure where exine is absent. For your competitive exam, all these details are very, very important. Intine is entire, exine is interrupted. Where it is interrupted? Where germ pores are present, there exine is interrupted. Now, what is the composition of exine and what is the composition of intine? We all know that pollen grains can be stored for several years. Do you know what is viability? Viability means it is a duration in which the pollen grains have ability to germinate and produce the pollen tube. That is viability. This viability will vary from plants to plants. In the later part, we will discuss in detail. Now, this exine made up of sporopollenin. Please remember this word, sporopollenin, which is one of the most resistant organic material now. In exam, all these will be matters a lot. Exine is made up of sporopollenin. Sporopollenin is a type of organic material. It is not inorganic. Organic material, and it can withstand high temperature and strong acids and alkali. Because of that, no enzyme that degrades sporopollenin is so far known. Illiya tanaka. That means till here, nobody had discovered the type of enzyme which have capability to degrade the sporopollenin, which is present on the exine wall of the pollen grain. Now, this pollen grain exine has prominent apertures called germ pores where sporopollenin is absent. So sometimes in exam, if they ask where the sporopollenin is absent in pollen grain, your answer will be on the germ pore region. Pollen grains are well preserved as a fossils because of the presence of sporopollenin. For many years, we can store them or preserve them. The only reason is because of sporopollenin, because so far there is no enzymes are identified which have ability to degrade this. The exine exhibits a fascinating array of patterns and designs. So if you observe the slides, here you can see a fascinating array, means arrangement of exines. So some of them are spiny, some of them are spiral, some of them have oval shape, some of them have depressions. So there are so many types of pollen grains are present. That is mainly due to the presence of exine. Okay, the exine exhibits a fascinating array. And why do you think the exine should be hard? Why do you think it should be hard? Because it is the layer which protects the pollen grain. And what is the function of the germ pore? Helps in the formation of pollen tube. Now coming to the inner wall. As I said, inner wall is made up of intine. Okay, inner wall is made up of intine. Now what is the composition of intine? It is made up of cellulose and pectin. Please remember, again a very, very important concept for competitive exam. The composition of exine is sporopollenin, which is organic, and here composition of inner wall, intine, which is entire. and it is made up of cellulose and pectin. The cytoplasm of the pollen grain is surrounded by a plasma membrane. That means intine is present exterior to plasma membrane. When the pollen grain is mature, it contains two cells, the vegetative cell and generative cell. This I explained. Once the pollen grains attains its maturity, the single nucleus undergo mitosis and produces the two cells. One is generative cell, one is vegetative cell. If you compare the size, generative cells are smaller, vegetative cells are larger, and generative cells, they floats in the cytoplasm of the vegetative cell. Why? How these two unequal sized cells are produced? By the formation of asymmetric spindles. That part is very important. The vegetative cell is bigger, has abundant food reserve, and large irregularly shaped nucleus is present. So this is the characteristic feature of vegetative cell. Sometimes while framing the questions, they just change the word. Here in case of giving vegetative cell, they just change it to generative cell. You should know the exact character. If you know the purpose why they have it, then finding out the solution will become very easy. See, vegetative cell is bigger. Why? Because it has to form a pollen tube. So to form a pollen tube, it requires a large amount of cytoplasm. So they have abundant food reserve, and a large irregularly shaped nucleus is present. Actually, it helps in guiding the pollen tube from the stigmatic surface to the egg. Okay. The generative cell, till micropylar region, it will guide, and later on they are accepted by the filiform apparatus that we will study in pollen pistil interaction. The generative cell is small and floats in the cytoplasm of the vegetative cell. Again, this statement is very, very important. Most of the students, they find it very difficult to understand how the generative cell floats in the cytoplasm of the vegetative cell. Hope my explanation made you to understand this very clear. Okay. So it is spindle shaped with the dense cytoplasm and nucleus. Spindle shaped nucleus. Okay, that is the characteristic feature of generative cell. In over 60% of the angiosperm, pollen grains are shed at 2-cell stage. That means remaining 40% of the plants, they shed their pollen grains at 3-cell stage. Why this mechanism is there? Then what happens in 60% of the plants? Whether they produce 2 male gametes? Of course, yes. When the generative cell, they float in the pollen tube, at that time, when they travel half of the distance, this generative cell again might undergo mitosis and produces the 2 generative cells. Otherwise, remaining 40% of the plants, what happens? Even before pollen grains are released, they become 3-celled pollen grains, where it contains 2 generative cells, which is nothing but the 2 male gametes. Okay, but what happens in 60%? These generative cell undergo mitosis in pollen tube. So the remaining species, generative cell divides mitotically to give rise to a true male gametes before pollen grains shed. Okay, so remaining 40%, before the pollen grains are shed from the anther, they form a 2 generative cells, nothing but the 2 male gametes. So pollen grains are the gametophytes and generative cell are the male gametes. That is very, very important. Now, pollen grains of many species cause severe allergies and bronchial afflictions in some people, often leading to a chronic respiratory disorders, asthma and bronchitis, etc. So where do we observe this? Sometimes, if your house is near to any plantations like acacia, or your house is near to a place where the certain type of weeds are present, like Parthenium grass, they produce the pollen grains. They are very minute, so they are present in the air. When you inhale, along with the air, these pollen grains will enter your respiratory tract and they severely cause allergies. Continuous exposure of individual to these type of pollen grains may lead to asthma or bronchitis. You know this, these two are the disease related to our respiratory tract. Okay, there is a difficulty in breathing, individuals who are suffering from asthma and bronchitis. It may be mentioned that Parthenium or carrot grass that came to India as a contaminant with imported wheat has become ubiquitous in occurrence and cause pollen allergy. Just now I took this example at the time of Great Famine. So in the time of 1947, when we fought freedom, India was in great danger because of famine. You know, there is a lot of competition for having food. So India imported wheat from British countries, and at that time, as a contaminant, this Parthenium grass entered India, and now they are spread all over the place. You know, any exotic plants, unscientifically, if they are exposed to any area, there are no predators for those organisms, so they establish very well, and they cover most of the surface of the land and fertile soil. Okay, so and these carrot grass is one of the headache for farmers because it's a very massive weed, and their pollen grains used to cause allergies in farmers or the individuals who get exposed to these pollen grains. Now, apart from this bad effect of pollen grains, now coming to the positive effects or benefits of pollen grains. See, pollen in some countries, it's very passionate that to have a pollen grains as a source of food. So they take pollen grains as a tablets, as an energy source. You know, the major diet of honeybee is pollen grain actually. They just conserve the nectars for off season, but on season when the flowers are, they take the pollen grain as a source of their food. Okay, so pollen grains are rich in nutrients. It has become fashion in recent years to use the pollen grain as a source of food supplement, and in Western countries a large number of pollen products in the form of tablets and syrups are available in the market. So these are some of the pictures. Here you can see there are some box where the pollen grains are stored, and here you can see the tablet of pollen grains. There is one sachet which contains 30 tablets. You know, in some countries, these athletes, just before their event, they used to take these pollen tablets. It is assumed that by taking pollen grains, their energy will be enhanced. So pollen consumption has been claimed to increase the performance of athletes and race horses. That's why in some countries they use the pollen grains as a source, great source of food. Now, once they are shed, the pollen grains have to land on the stigma surface. Now, what is the primary function of the pollen grain? It's a male gamete. It's a male gametophyte carrying male gamete. Now, it has to deliver the male gamete to the place where egg is present, where fertilization takes place. So, how long do you think the pollen grains retain viability? Already I explained you. Certain pollen grains have viability for a day. Certain pollen grains, they have viability for months. Certain pollen grains, they have viability till month. Okay, here some examples are given. Period for which pollen grains remain viable is highly variable, and some extent it depends on the prevailing temperature and humidity. See, this is very important. Even seeds also impacted by temperature and humidity. In the same way, pollen grains are also impacted by varying temperature and humidity. If high temperature is there, pollen grain may lose viability early. If the favorable condition is there, the viability may extend up to some time, or if humidity is high, it will change. So these factors, environmental factors, also influence. But in some cereals such as rice and wheat, pollen grains lose viability within 30 minutes. Again, these are some very important concepts. MCQs may be formed on these concepts because in competitive exam, they go through these type of sentences where the factual data is given. Example is cereals like rice and wheat, pollen grains have viability up to 30 minutes. As soon as the flower shows anthesis, when the flower blooms, pollen grains have viability just for 30 minutes. Before that, it has to reach the stigmatic surface and pollination should take place. Okay. And in some members of Rosaceae, Leguminosae, Solanaceae, you know Rosaceae, rose plants are present, Leguminosae, like beans are present, and Solanaceae, brinjal, capsicum, tomato, all these plants maintain viability for months. For months, these pollen grains, they maintain the viability. You may have heard of storing the semens, sperms of many animals, including humans, for artificial insemination. Now, there is a reason to tell this example, how the semens are stored. You know, this process is called cryopreservation. That terminology is not given. Here they have directly given the pollen grains are stored in minus 196 degrees Celsius. That process we call it as cryopreservation. In cryopreservation, in liquid nitrogen, the temperature of the liquid nitrogen is minus 196 degrees Celsius. These male gametes like pollen grains and sperm in the semen can be stored, which later can be used in pollination, or in the case of animals for insemination. So such stored pollen can be used as a pollen banks, similar to seed banks in crop breeding programs. A very, very important aspect. See, genetic resource of any plant is very important for any country. So sometimes what happens, because of over exploitation, plant may undergo complete extinction. How do you conserve the genetic resource of such plants? You can store their pollen grains in pollen banks, and whenever it is required, you can go for the tissue culture where you can produce the new plants. Or if the seeds are not viable, then you conserve the pollen grains and use the pollen grain in tissue culture and generate the new plants. So the main importance of cryopreservation is you can store these pollen grains for several years. Okay, so that is about the pollen grains.